Rare diseases often remain undiagnosed and untreated due to unknown etiologies. In recent years, researchers have made headway characterizing the molecular causes of different rare conditions, thanks to progressively powerful genetic methods such as whole genome and exome sequencing. As it becomes faster and more accessible to find causative mutations, scientists look toward the future, envisioning a world with earlier interventions and the possibility to stop rare disorders in their tracks before their symptoms even appear. Welcome to The Scientist Speaks, a podcast produced by the Scientist Creative Services team. Our podcast is by scientists and for scientists. Once a month, we bring you the stories behind newsworthy molecular biology research. This episode is brought to you by Valsera, a company dedicated to advancing precision medicine through innovative sequencing solutions. Valsera understands the challenges faced in diagnosing and treating rare diseases, and their expertise lies in providing cutting-edge tools and technologies to accelerate research in this critical area. By harnessing the power of next-generation sequencing, Valsera is committed to empowering scientists and clinicians to unlock the genetic underpinnings of rare diseases. Together, we can work towards a future where every patient has the opportunity to receive an accurate diagnosis and effective treatment. In this month's episode, Deanna MacNeil from The Scientist spoke with medical geneticist and chair of the Department of Pediatrics at Boston Children's Hospital, Wendy Chung, to learn how the rare disease field has evolved alongside advances in next-generation sequencing.
Nearly all human diseases are influenced by genetic variation. But which genes are the culprits behind heritable predisposition and pathologies has not always been tractable knowledge. Although researchers began putting the pieces together on heredity and genetics in the mid-19th century, it wasn't until the 20th century that an elucidated DNA structure and code, as well as readily available genetic technologies, helped researchers gain ground on disease heritability. Wendy Chung's career has advanced in step with the steady progress of genetic breakthroughs since the expansion of laboratory techniques such as Sanger sequencing and PCR. As a trailblazer in rare disease research and treatment, she credits her current passion for capturing the big picture of rare disease genetics with sequencing to her time as an undergraduate biochemist.
About 35 years ago, I started working at the National Institutes of Health in the laboratory of Seymour Kaufman on a condition called phenylketonuria. That condition is what we call an inborn error of metabolism. That was my first real introduction to genetics when we were sequencing people that had abnormal or atypical forms of phenylketonuria. And we started to identify their mutations in genes that were not the typical genes we would see for phenylketonuria. That started my love affair of understanding not just biochemistry, but genetics, and that blossomed into really thinking about not just one gene, but the whole genome.
The sequencing methods that Chung performed while studying phenylketonuria, or PKU, are not what many scientists might associate with the concept of sequencing today. At the outset of these methods, dideoxy, or Sanger sequencing, was the holy grail of genetic techniques. Researchers would clone genes of interest into plasmids and express them in bacteria before performing gel electrophoresis on radiolabeled DNA to identify each nucleotide in a gene and visually pick out any differences between individuals. Sanger sequencing's accuracy, robustness, and ease of use contributed to it becoming the most common DNA sequencing method for decades. But as with many molecular techniques at the time, the lengthy and manual nature of the workflow was a limitation.
Some scientists or graduate students will laugh about this, but we used P32 radioactivity. We ran it out on these very thin glass plate enclosed polyacrylamide gels and manually using light boxes and being able to look at these dark bands on X-ray film and read out the sequence. It was an incredibly laborious process. It would take me months, potentially, to sequence a patient's entire gene in this way. Things like polymerase chain reaction or a molecular Xerox machine was advanced in the very early days when PCR was first available. I remember as a graduate student sitting there, and I was the human thermocycler, taking tubes and moving them from ice buckets to heating blocks to room temperature, moving it between those three stations over hours. So laborious, incredibly boring. Thankfully, someone invented the thermocycler that did that automatically and made my life easier as a graduate student. There were methods that were then advanced to use fluorophores instead of radioactivity, and that was definitely good for our health. People found ways of being able to use capillaries as polyacrylamide to put the sequencing data through, and that became much easier than pouring these big slab gels. People developed informatic methods to be able to read this out in an automated way rather than reading films manually. But even still, all of those were like child's play compared to what became so-called next-generation sequencing.
Next-generation sequencing, also called NGS or second-generation sequencing, includes several high-throughput approaches that involve massively parallel processing. Rather than cloning individual genes into plasmids, NGS begins with a DNA sequencing library made of many amplified gene fragments. Unlike chain termination labeling in Sanger sequencing, the DNA is sequenced by adding nucleotides to complementary strands, and spatially separate DNA templates are sequenced simultaneously without a physical separation step. The advent of NGS methods allowed scientists to rapidly sequence full genes on a larger scale, which has revolutionized disease research. Because these approaches are less labor-intensive and generate large volumes of sequencing data per run, they enabled researchers to identify many causative rare disease genes and variants for the first time, and helped dissect multifactorial genetic traits through examining entire genomes and population-level variability.
When I first started this, we knew very few of the genes that were actually responsible for human diseases. The first big step forward was to be able to positionally clone or identify essentially the address where a gene lived, and then by narrowing down the address, identify the actual gene itself. The first gene that I identified that way took me 8 years. The last gene that we identified took less than 8 days. By knowing the roadmap, by having the blueprint of the human genome, having all of this information laid out, it allowed us to interrogate the entire genome in one fell swoop, and in some cases within hours or days. Putting that together, though, the first time, essentially what was the Human Genome Project, took quite a while. When I started the first year of my MD PhD program was the first year the Human Genome Project was announced. When I started within this field, we didn't have that blueprint. We didn't have all that information, and hence the reason why it took me so long at the beginning to be able to identify these genes for diseases. But now that we know most of the genes, maybe not all, but probably most of the genes that humans have, being able to make that association about what gene is responsible for any one patient's disease becomes very rapid. Even if that gene has never been implicated in a human disease before, it becomes a much easier task to do. And of course, for me, ultimately, the goal is not just diagnosis, but improving lives, being able to use that information to make improvements for patients beyond just giving a name to their symptoms.
The Human Genome Project was part of a concerted research effort to progress from studying one gene at a time, nucleotide by nucleotide, to investigating many whole genomes in parallel. This opened new opportunities in population-level genetics research, allowing scientists to screen for rare mutations or variants more easily, and transforming a process that was once like finding a needle in a haystack into one that was more like spotting the difference between a blade of grass and a straw of hay. As NGS techniques continue to enable this unprecedented era of disease gene identification in the laboratory and the clinic, scientists like Chung have their eyes on the future of sequencing-enabled therapeutic development, finding actionable avenues in human genetics research. In some of her research team's latest translational work, Chung tapped into that potential for a rare and degenerative neurodevelopmental condition.
We work on a number of conditions associated with neurodevelopmental disorders, epilepsy, intellectual disabilities, and in many cases, trying to characterize what we call the natural history or how those conditions evolve over the life course. One example is that I work on a condition that's called KIF1A associated neurological disorder, or CAND for short. It is a condition that's associated with epilepsy, intellectual disability, developmental delays. And it's a particularly urgent condition that I study because it's a degenerative condition. In other words, you lose ability over time. And for some individuals with CAND, it's ultimately lethal. We've gotten very good at recognizing that condition, making the diagnosis, being able to a certain extent, predict the prognosis because there's a wide range of different mutations and different prognoses in that gene. But for me, one of the most gratifying things has been developing a treatment for that condition. We've developed what's called an antisense oligonucleotide that we can inject into the space that eventually gets to the brain, into the spinal cord, to be able to treat that condition. And although we've only treated one person so far, that treatment has proven to be successful in terms of slowing down the disease and improving some of the symptoms. While it's still only one person and it's still only a little over a year's worth of treatment, it suggests that for that condition, we may be able to change the trajectory in the future for people with that condition.
Antisense oligonucleotides, or ASOs, are gene therapies that bind to specific target RNA sequences and modulate downstream protein expression. Because ASOs target disease drivers at the RNA level, they can correct pathologies at one of the earliest actionable steps in a genetic pathway. In the case of CAND, this N=1 clinical study targeted a patient-specific mutation in the KIF1A gene and slowed down disease progression. Although it may not be the solution for every patient with CAND, Chung's success with the ASO treatment is a hopeful compass pointing in the right direction. The next step becomes easier and the path clearer with the help of genetically guided scientific rationales. For instance, researchers can use established strategies like ASOs to determine which other conditions may be candidates for similar genetic therapies. In the case of CAND, there is much work ahead. Researchers do not yet know if ASOs will work for other patients, but Chung hopes that rinsing and repeating this scientific process may yield a proverbial light at the end of the tunnel for more individuals. In addition to helping determine the right type of treatment, sequencing technologies contribute to earlier rare disease diagnosis for potentially better therapeutic windows. Chung estimates that about 500 individuals around the world have been diagnosed with CAND, but there are likely many more people affected who haven't had sequencing to know the name of their condition. Improving access to genetic diagnostics may create opportunities for earlier interventions, not just for CAND, but for other rare disease types that have historically faced diagnostic hurdles.
Some of these conditions, for instance, require treatment within the first year of life. Many patients with these rare diseases are not getting diagnosed until they're teenagers. A couple weeks ago, I diagnosed one of my patients that, although there's now a genetic therapy, she wasn't diagnosed until she was 72 years old. She has a diagnosis of Rett syndrome due to a mutation in MECP2. She's had that her entire life, but when she was born, we didn't even know that was a condition. When she was growing up, it couldn't be diagnosed because we didn't know it was a thing. Needless to say, we didn't have the technologies from a genetic point of view. For many adults, they have quit looking for a diagnosis from a genetic point of view. They know their symptoms. They, at this point, have found ways to adapt or to cope with their symptoms, and so they don't look for a genetic diagnosis. It doesn't happen very often that I get 60, 70-year-olds coming to me for diagnoses of those types of symptoms, but we can make them on individuals of any age. I think the opportunity, as I think about it, though, is for the next generation growing up, knowing that there is now one FDA-approved treatment for Rett syndrome, knowing that there are gene therapies that are in clinical trials for Rett syndrome. I look towards a future perhaps where you could diagnose those girls at birth and maybe prevent what would be a lifetime of disability.
As scientists become more adept at implementing NGS methods, that future may soon be within reach for a multitude of rare diseases. Chung and her research team are now piloting a consented research study called Guardian. an acronym for Genomic Uniform Screening Against Rare Diseases in All Newborns. For parents who join the study, over 450 genetic conditions will be assessed through genomic sequencing. The researchers can sequence newborn DNA using only a tiny blood spot from a heel prick that is already collected for standard newborn screening, a test with connections to Chung's research roots in PKU genetics.
We've gotten good at using small amounts of DNA. Even just a couple drops of blood is all you need to be able to sequence a genome now. Being able to do genome sequencing on the heel prick that we use for newborn screening that was started for phenylketonuria, using that dried blood spot that we use for newborn screening and generate DNA sequence data on many, many more conditions than just PKU. Conditions where there's news you can use, there's information that is important for treatment. Being able to have that diagnosis even before the recognition of symptoms or the onset of symptoms may allow us to start a treatment, to do something that will improve the health of the next generation. We're still researching this to understand feasibility and how to improve it, to optimize it, to get it to the point where you could do it on scale. But I think the cost of sequencing is decreasing low enough, and our ability to interpret and understand the genome great enough that for some conditions, this could have benefit to do on a population scale. Things are changing rapidly, so what I said today may be very different in a year. I actually hope that's the case because I think there's a lot of potential. I'm a physician and a scientist, but it always starts with the patient and ends with the patient. All the things that we think about are always to improve the lives of our patients and their families, and that brings me a lot of joy.
Thank you for listening to The Scientist Speaks. This episode was produced by the Creative Services team for The Scientist and narrated by Deanna MacNeil. Thanks again to Valsera for sponsoring this episode. Please join us again in August as we learn about the influence of age-defying benefits of fasting. To keep up to date with this podcast, follow The Scientist on social media and subscribe to The Scientist Speaks wherever you get your podcasts.